how, I forgot how much the bonus gonna be worth.
Huh?
The bonus, the project.
The project 50, if you do that. Okay, so last time we were talking about the patterns of island biogeography, colonization and extinction, and how we have an equilibrium between colonization rates and extinction rates that tells us the average number of species we can expect to find on an island. But we have to go through these rates just a little bit because people often mistake what they mean. So as soon as I do this, we'll get to that. All right, so what do we mean by these rates? It's critical to understand this in order to interpret what's going on. Well, what you need to do is to imagine that you have an island that's empty. And there's a source, maybe a mainland, that has a bunch of species. And so the way you interpret these rates has to do with the arrival or extinction of species on the island. But what we're talking about here is, in the colonization curve, the arrival of new species, not the arrival of any species. It's just new species. And that's the key to understanding why this curve goes down. So let's imagine that we have a mainland here, and it has 50 bird species. And here's a little island that's empty. All right, so we have then some birds that arrive, okay? Since the island is empty, what's the frequency of arrival of new species? The rate at which new species arrive on this empty island. If there are no species on the island, and you get one new species arriving, what's the rate of arrival of a new species? 100%, one, yeah. So that right there symbolizes zero species, an empty island, and this is one. This is the rate of arrival of new species when there are no species on the island already. Now, this rate has to go down because once you get one species on the island, only 49 out of the 50 would be new species. So here you have one species on the island, and only 49 of the 50 are new. So the rate falls. If you have 10 species on the island, then only 40 of them could be new arrivals. Getting an arrival from a species that's already on the island does not change the number of species on the island. Okay, and when you wind up with 50 species on the island over here, every species that could possibly arrive from the mainland is already on the island, so no new species arrive. So the thing to focus on here is not the rate of arrivals. It's whether the arrivals represent a new species. If you have a bunch of species on the island already, then very few of the dispersers that arrive are going to represent a new species. So this is the colonization rate of new species on the island. So it's important to understand that rationale. What we're comparing here is the rate at which new species arrive. That's going to be 100% when there's nobody on the island. And when everybody on the island represents the species that are already on the mainland, or when the species on the mainland are represented on the island completely, then it's not possible to get a new species. because the species are all already there. So this colonization curve declines as the number of species on the island increases. Okay, do we have questions on this? Because it's actually quite important in understanding this. All right, well, why would the extinction rate increase? Well, for one thing, if you have 0 species on the island, how many species could possibly go extinct? 0. If you have 50 species on the island, how many species could potentially go extinct? 50, right. So this has to be an increasing curve in some fashion. Now, there's an additional set of circumstances that causes that extinction rate to go up. Not only do we have kind of the trivial idea that if you have 0 species on the island, there's 0 possibility of extinction. If you have only 1 species on the island, how much competition does it experience from other species? None. It has the whole island to itself. So if you have no interspecific competition, what does that do to the resources that each individual is likely to get? Should be pretty high. Species that have little interspecific competition should have relatively high individual fitness. That means they'll have a lot of resources for reproduction. That means that they'll be able to maintain a large population. We know something about large versus small populations, right? Which are more likely to go extinct? Large or small? Small are more likely to go extinct just from random stochastic events. So now consider if we have 10 species on the island, they're dividing up the resources. Each individual of those species experiences both inter and intraspecific competition and gets fewer resources than if it was the only species on the island. So what does that do to the individual fitness? Rees says it decreases individual fitness. What would that do to the reproductive rate then? It would decrease it, yes. So what would that do to the effective population size of each of those 10 species compared to if there was only 1 species? Smaller. So as the number of species on the island increases, it's not just this that there's this trivial, if you have no species, you can't have any extinction, and if you have 50, you can have a lot of extinction. There are biological processes pushing a higher extinction rate. Competition for resources increases, and as interspecific competition increases, individual fitness goes down, reproductive rates go down, the population size goes down. This is one species that has a large population because it has the island all to itself. Here are 50 species that are competing intensely for resources that have small populations. So just from competition for resources and having smaller populations, you're going to have not just 0 versus something, but 0 versus a really steeply increasing extinction rate. So you need to understand what the underlying logic of this graph is. Okay, the more species you have on the island, the fewer arrivals from the mainland are going to be new species. The more species you have on the island, the higher the level of interspecific competition, the smaller the effective population size, the higher the likelihood of extinction. So the rate at which new species arrive crosses the line at which existing species go extinct at some point, and that tells you, on average, the number of species you can expect on that island. Now notice we haven't said anything about which species, and that's really a crucial prediction of island biogeography theory. The number of species on an island is going to remain approximately stable, but the specific ones that you find at a given time may differ from the specific ones that are present at a different time. Because just randomly, a species that was on the island may go extinct, and it may not be replaced by the same species. It may be replaced by a different one. So in island biogeography theory, the crucial thing here is this S hat, which is the stable expected number of species on an island, but the identity of those species is not fixed. It can be any of the species in the pool. So let's look at how now island size and island distance should be expected to affect these things. Okay, so here's island size, and here's the colonization rate. So what you're thinking of here is two islands that differ in size that are identical distances from the mainland. There's a small one, there's a big one. So colonists get out to these islands at the same rate. So the colonization rate is the same. The arrival of new species is identical on these two islands. However, the large island supports larger populations, so the extinction rate is relatively low. The small island supports small populations, and the extinction rate is high. So when you look at where the lines cross, this would predict that the large island would have, on average, more species than the small island. And it is because of the difference in extinction rate. Okay. What about island distance? Now let's imagine two islands that are identical in size, but differ in distance from the mainland. Well, that's bigger, but you get the idea. Here, I'll try to make it simple. Okay, hey, who says I can't draw? Besides you guys. Okay, now there is a close island that is easier to get to, so the colonization rate is higher. You have a far island that is harder to get to, so the colonization rate is lower. So here's the higher colonization rate, nearer the mainland. Here's the lower colonization rate, away from the mainland. The extinction rate is identical because they are the same size. They offer roughly the same opportunities. They support populations of the same size. So the extinction rate is identical, but you get more species arriving on the near island than on the far island. So the equilibrium number of species near is higher than the equilibrium number far. Okay, it's really important that you guys try to look at those two graphs and try to understand the relative importance of near versus far and large versus small and which curve they affect. Because I guarantee you that when you see this on the exam a week from today, oh dear, you will screw this up unless you pay attention to these graphs. Okay, well, we can then use this in an archipelago like Indonesia, and we can make a whole bunch of predictions about which island should have more, which island should have less. It can get messy, and here's something to kind of remind you of maybe what the real food web looked like. It was way too complicated. This is way too complicated. So here are large islands, and here are small islands, and here's progressively higher extinction rates as you get smaller and smaller islands. Here's near islands, and here's far islands, and progressively lower immigration rates or colonization rates. And so you can predict a whole variety of equilibria there. We don't worry about stuff that's quite this complicated in this class. We're just interested in the basic effect of distance on colonization and the basic effect of size on extinction. Now, this is great. Okay, we have this beautiful theory. E.O. Wilson and Robert MacArthur wrote a book about it. It was a bestseller. It sold all of like 200 copies because they were selling to ecologists who don't have a lot of money, and there aren't a lot of them. But it's one of the most influential books ever written in ecology. But this is great. We have these predictions. Do they actually work? Do they actually predict anything? Well, okay, so you have to do experiments to actually verify predictions, right? How do you do experiments on entire islands? Well, there are natural experiments like the eruption of Krakatoa in Indonesia in 1883 that kind of wiped out all the life on nearby islands, and we were able to see them recolonized over time. But, you know, that has the same problems that all natural experiments do. They're not controlled. There's all kinds of stuff going on. We might see patterns for one reason or another. What we really need is a manipulative experiment. Okay, so I'm going to show you a manipulative experiment that is kind of the gold standard for understanding island biogeography. And I'm only going to show you part of it because it's much more complicated than this. All right, well, E.O. Wilson is perhaps the greatest naturalist of the second half of the 20th century. He's a professor at Harvard, and he's had all these students. He's written all these books. He's the guy who invented sociobiology. He has like 800 publications on ants and societies. But he also had a lot of good graduate students. One of his graduate students was named Dan Simberloff. And Dan Simberloff came to Harvard just after he wrote that book. And he said, well, that's great. Can we test it? And Wilson said, I think I have an idea. So they went to the Florida Keys. And the Florida Keys, for those of you who don't know, consist of like 10,000 tiny little islands. And some of them are this big. So here's a person standing on a scaffold. Here's another person. That's an island. So we're not talking about big islands, right? But what they did was count the number of species on several islands off the coast of Florida at different distances and different sizes. And then they erected this scaffold, put a tent over the island, and fumigated it to kill all the insects. And then they asked, well, if island biogeography theory is correct, then we should see an approximate return to the same number of species that we had before our treatment, even though the specific species might not be identical. And so, here's the number of species that originally were on the farthest island. So we're just going to look at distance, not size, in this little graph because it's complicated enough. Here's the number of insect species on the intermediate distance island and on the the near island. Okay, so here's fumigation day, and here's the recovery trajectory. Okay, notice that it's not exact, but the order is the same. The order is as predicted. The near island had more species than the intermediate island, and the intermediate island had more species than the far island. And that's exactly the order in which they originally were. Now, you can't make too much out of this failure to achieve the pre-treatment numbers of species because the weather affects these things, and we don't know how much time it really takes for this to occur. So the important thing is that we got the results in terms of number of species that we expected. We got the other result as well. It was different species that recolonized. Some of the same species got back to those islands, but some of them didn't, and they were replaced by other species. The species number was stable, but the species identity was not. That's exactly what we predict from island biogeography theory. Now, a word to the wise, don't approach the U.S. Park Service about doing this again. This is now the national park. This is the Biscayne Bay National Park. They take a very dim view of eradicating all life on trees in Biscayne Bay. So you can't do this experiment anymore. However, back in the 1960s when this was done, the Park Service had a little more liberal attitude towards this. And they said, oh, sure, why not? They don't do that anymore. Just like they don't allow you to kill all the birds in a forest to establish whether you're looking at character displacement or simple competition. Okay, so we in fact do have experimental confirmation of island biogeography theory. Species numbers are conserved, species identities are not. The interesting thing about island biogeography theory is that you can apply it to any kind of island. So these are islands of trees in the middle of the ocean. But there are lots of other kinds of islands. Here's the state of Nevada. And what you see here is mountain ranges. So all of these things here trending north-south, those are all isolated mountain ranges. They are all separated from each other by intervening desert valleys. They are functionally islands. They're alpine islands in a sea of desert. Well, the same considerations then should apply as apply to islands in the actual ocean. So we can look for species that occur at high elevations and ask whether size and distance from the mainland source of species predicts the number of species that you'll find on the islands. So here is a really large island. It's pretty isolated though. It's in the middle of Nevada. Where are the mainlands? The Sierra Nevada mountains in California is one mainland. There's another mainland way over here. That's the Rocky Mountains on the far side of Utah. So this is a long way from anything looking like a mainland. So here's a very large one. Here's a very small one. There's another small one. Here's another really large one. So size and distance should predict what kind of species or the number of species that you get on these islands. Furthermore, the numbers should be predictable. The exact species should be less predictable. So what do these sky islands look like? We call them sky islands. And this is what they look like. This is the top of the mountains. And this is the desert that separates them. And you see wild horses here. That's lovely, but kind of irrelevant. The species that live here cannot live here. So it is exactly analogous. So what patterns do we see when we look at these sky islands? Well, okay, here's the mammal species, and here's the area of the sky island. Well, hey, that's exactly what we predict from island biogeography theory. Here's the number of mammal species and the distance in kilometers from the mainland. And that's exactly what we predict from island biogeography theory. Okay, we don't put the colonization and extinction rates together on a single graph for this data because it's pretty confusing. It's real data. It's not just those simple curves. But you can see that the number of species follows the prediction of island biogeography pretty exactly. So mammals in particular have a hard time getting across those desert stretches, And they are basically isolated on those mountain ranges from the last interglacial, the last glaciation, which changed the habitat in the Great Basin from sort of desert in the low areas to kind of tundra in the low areas. So they were able to disperse all over the Great Basin during the Ice Age. They then got isolated on the mountaintops as the weather warmed, and they lost species in the smallest islands, and they were not able to recolonize in the smallest islands. So we're following the predictions exactly, even though it's not a true island. Okay. Interestingly, birds follow the same pattern, but because they disperse better, bird communities are more similar across the Great Basin. The line here in terms of area is shallower, and the line here is also shallower, and it's kind of up here. So the ability to disperse makes a difference, but island biogeography theory is an excellent predictor of things that are really important in conservation biology, and it is one of the main foundations of conservation biology. That's because human activities have converted most habitats on Earth into habitat patches, separated by a matrix of unfavorable habitat. Whether that's farm fields or desert from human activities is irrelevant. We understand what to expect in patches of habitat regardless of where they are based on island biogeography theory. So this then brings us to a consideration of what we call landscape ecology, which is the application of these principles to habitat patches in general. So, let's, where are we here? Slideshow. So we will cover then what we consider to be landscape ecology. We'll talk about something that we haven't talked about yet, which is shape. The shape of a patch and edge effects, and these two things are related. We'll talk about the kind of gold standard for understanding patch shape, which is fractal geometry. And then we'll look at patch structure and movement patterns, corridors, and ecosystem engineers in landscapes. Remember, we talked about keystone species often being top predators. Here we're going to focus on things like beavers and elephants and how they alter landscapes. So landscape ecology is a fairly new branch of ecology. It was founded in 1980 with a publication by Monica Turner of the University of Wyoming that was entitled Landscape Ecology. So a landscape is simply a heterogeneous area consisted of patches. The patches may be separated by a matrix of unfavorable habitat. And landscape ecology is the study of spatial patterns and processes, ecological processes, across that landscape at a range of scales. So we are directly applying island biogeography when we talk about landscapes. And here's a great example of landscape structure and why it's important. So here are 6 landscapes. And you can see that we have small isolated patches here. Small isolated patches here. These are actually counties in New England. And here's some larger patches that are also isolated, and here are some really large patches. So interestingly, we can see that some of the patches are really long and skinny, Some of the patches are short and fat. And it's worth asking, like, what do you think the effect of patch shape is? So the total area in dark patches here is kind of similar. The total area in dark patches here is, kind of similar. This has a lot less. But if you're looking at these patches, compare Monroe at the top left, which is this one, and Hudson down here, and let's just see what you think. Which has more forest? Does Monroe have more forest than Hudson? Does it have less forest than Hudson? Does it have about the same amount of forest as Hudson? This is difficult for people to figure out. So it's a question really worth considering. And you'll see why we use quantitative tools rather than our impressions. So we're open, if you've got your clickers. And let's see. Five, 4, 3, 2, 1. Okay. So let's just check this out. What do you think? Is it this one or this one? Which has more, or are they about the same? Let's see. And got to do that. I've got to do this. Okay, so most of you are going for about the same.
What's the wrong?
I'm not going to tell you yet, because we're going to do one more question, which is harder. This is even harder. Yeah, whatever Ahmad wrote is wrong. Just you can count on that. So now you first were trying to compare. Now try to tell me what the percent forest cover in this one is. Is the percent forest cover about 1, about 5, about 10, about 20, or about 30? Fair warning, this is harder.
It's about 25.
Well, that's not an option. Whoops, hello, come on. That's not an option, you have to pick one. What's it closest to? Okay, 5, 4, 3, 2, 1, and closed. Okay, so there's some uncertainty here. I'm not going to make you argue about this. We're just going to tell you what it is, because you have no basis for actually comparing. And so most of you think it's somewhere around 20%. Okay. These things are notoriously difficult to figure out. And we cannot rely on our impressions for this. So if we actually look at what's going on here, this is Monroe and this is Hudson. Here's Monroe and this is Hudson. They are almost identical, these two. So the answer to the first one is that they're about the same. But look, this has fewer but larger patches than that one. That's important. Somerset here is right there, and it's about 20%. So more of you got 20% than I would have predicted. I would have predicted a lot more thinking it was less and a lot more thinking it was more. Not a bad job. Okay.
I told you it was around 25.
What's that?
I told you it was around 25, though.
I'm not judging. I'm just going through the exercise to show you how difficult it is and why we use these tools. Okay, so I'm going to skip a couple of slides here, and whoop. There we, yes, they are, and we don't have time for them. So here's something about that comparison between the two counties that had identical, more or less, proportions of forest. One had a lot of scattered small patches. One had a few big patches. That makes a tremendous difference. Oh, and we may or may not have another question. The reason that it makes a difference is because of what we call the edge effect. So here's a patch, and here's the edge of that patch. Surrounding the patch is something other than the habitat. So let's think about this. Let's think about this as a forest and this as a field. The field is hotter. It's brighter sunlight. It's drier. Well, those conditions penetrate the edge of this patch. And they actually reduce the area that is effectively forest. You may see trees out here, but it's not a forest. It's an edge. The edge effect reduces the amount of habitat available for these core interior species. So maybe deer might prefer the forests to the field. Well, they have less space than you think they do because of this edge effect. Edges commonly extend 50 to 100 meters into the adjacent habitat. So there may be forest patches in New Orleans that are not, in fact, at all forest. They may have trees living there, but they may not provide forest environmental conditions anywhere in that patch. This is crucial. This is why island biogeography helps us here. To see the effect of the edge, let's compare. So here's a single large patch, and here are 4 small patches of equal size. But notice that the edge whacks off an incredible amount of these 4 patches, and so the interior that's available for forest species is much smaller when you have small patches than when you have a single large one of equal size. Patch size matters tremendously. Okay? This is, you may have the same area here, but because of the edge effect, you have much lower functional size. So when we look at the conservation value of patches, we pay a lot of attention to size. Okay. Now, how do we measure that edge? Because the edge is crucial for understanding what the effective size of a patch actually is. Well, okay, we're going to introduce you to another advanced math technique, which is fractal geometry. Fractal geometry is just the idea that if you use a ruler of a given size and measure the perimeter of a irregular shape, the perimeter that you measure is going to depend on the size of the ruler that you use. Well, here's the outline of England, and here we are using a ruler of 200 kilometers. So each of these little segments is 200 kilometers. Okay, and you might say, well, that's not a very efficient way of measuring England, but we're going to prove a point here. So this is connecting areas on the coastline that are 200 kilometers apart. And notice that we're missing a lot of indentations here. Okay, if we use a smaller ruler, we can get some of those indentations accounted for. If we use a smaller ruler still, we can account for more. If we use a actual 12-inch ruler, we'd account for virtually all of them. But look at what the perimeter looks like, depending on that ruler. Here, if you use a 200-kilometer ruler, you get a coastline of 2,300 kilometers. So you get an edge effect that is based on 2,300 kilometers. If you use a smaller ruler, you get a bigger perimeter. This is basically a measure of how irregular the shape is. If you have a perfectly round or perfectly square shape, then the fractal dimension, the rate at which the edge changes, is very small. If you have a really irregular shape, the rate at which the perimeter changes is really large. So you can graph the size of the ruler against the change in perimeter, and you can get a measure of irregularity. That turns out to be a really good predictor of how valuable a patch is, aside from its size. So perimeter and size are both important. Here's what we do if we actually show you what that graph might look like. So here's using a ruler length of 1 kilometer. You get a long coastline for this island. Here's a ruler length of, say, 40 kilometers. And notice that you get a much shorter coastline. Duh. What's important is the slope. The slope is what we call the fractal dimension, and that tells you how the coastline changes or the edge changes as the size of your ruler changes. It is a measure of how irregular this patch is. The higher the slope, the faster it decreases, the more irregular this shape is, and the greater the effect of edges is. So we look at fractal dimension as a measure of shape and irregularity. We look at size. Just because you have a big patch does not mean it's a great patch. If it has a high fractal dimension, if it's a really irregular patch, it may have very little value because it may be all edge. So those two things go together. All right, let's see. I'm going to skip that, and I'm going to go here and look at landscape patterns and animal movements. Okay, so we have landscapes that have fragmented patches. Here's a mountain range. These guys like mountains. In order to cross from one mountain range to another, though, they have to go through unfavorable habitat. The more heterogeneous a landscape is, the more difficult it is for animals to move because they have to cross that unfavorable habitat. That has real consequences for conservation. If you have a large block with a regular shape supporting a big core area, you're going to have a nice, big, healthy population. And they don't have to cross anything to find mates or find food sources. If you have a series of small, chopped up patches, the number of individuals is going to be much smaller for a couple of reasons. They'll have to move more, they'll have to expend more energy, and they'll be chopped up into a bunch of isolated small populations that will have a higher rate of extinction. We can show this experimentally. Here's a little experiment that actually a friend of mine did in Colorado, in the grasslands outside of Denver. She put rat traps on a grid in large patches and medium patches and small patches. And then she asked, well, how do the rats move in this environment? And here's how they move in that environment. So the average distance that they moved decreased as patch size increased. In large patches, they don't have to move much because they have everything they need. They have mates. They have access to areas of food, even if they kind of eat out one part of that patch, they can just move down the road to another. If they're in a small patch, it's easy for them to destroy the value of that patch, in which case they then have to move. They have to move longer distances to find another patch. They also don't have a lot of choices in terms of mates, and so they may have to disperse much longer distances. Okay. It's also easier for animals to move in larger patches. So they don't move much, but when they need to move, they just move. When these guys run into trouble, they find it difficult to move. And so they have to go a long way, and very few of them wind up being able to do what they need to do. It's a double whammy. It's hard for individuals in small patches to get what they need. They have to go a long way to do it sometimes, and it's very difficult for them to do that. If they can't do that successfully, for instance, if they eat all the food in that little patch, they have to go a long distance to find more food, and not many of them can find more food. What happens to the individuals that could not make that move? They die. Exactly. Okay, I've got a couple of more things to say about landscapes before we talk about global patterns of diversity. That will conclude the material for Monday's exam. So we will finish that on Wednesday. We will finish community ecology on Wednesday. On Friday, we'll start talking about ecosystems, but that won't be on the exam on Monday. Okay?